Welcome to our exploration of the history of genomics, a field that has revolutionized our understanding of life itself. Genomics is the study of genomes, the complete set of DNA within an organism, including all of its genes. This journey will take us from the earliest discoveries about genetic material to the cutting technologies shaping our future. We'll see how curiosity, collaboration, and innovation have driven scientists to unlock the secrets of our genetic code. Let's begin by setting the stage for this incredible scientific adventure and understanding why genomics matters to everyone. Long before the word genomics existed, people wondered how traits were passed from one generation to the next. Ancient civilizations speculated about inheritance, but it wasn't until the 19th century that scientific theories began to take shape. Gregor Mendel, an Austrian monk, conducted experiments with pea plants and discovered the basic laws of inheritance. His work laid the foundation for genetics, showing that traits are inherited in predictable pattern. These early insights set the stage for future discoveries about the molecular nature of genes and heredity. Next major leap came with the discovery of DNA as the molecule responsible for heredity. In the early 20th century, scientists like Oswald Avery, Alfred Hershey, and Martha Chase provided evidence that DNA, not protein, carried genetic information. This breakthrough shifted the focus of research and set the stage for understanding how genetic information is stored and transmitted. The identification of DNA as the genetic material was a pivotal moment, opening new avenues for exploring the structure and function of genes. In 1953, James Watson and Francis Crick, with crucial contributions from Rosalind Franklin and Maurice Wilkins, unveiled the double helix structure of DNA. This elegant model explained how genetic information is copied and passed on. The discovery of the double helix was a turning point, providing a visual and conceptual framework for understanding genetics. It also inspired a new generation of scientists to investigate the molecular mechanisms underlying heredity and gene expression. Central dogma of molecular biology, proposed by Francis Crick, describes the flow of genetic information. DNA makes RNA and RNA makes protein. This concept clarified how genes direct the synthesis of proteins, which perform most cellular functions. Understanding the central dogma was essential for the development of molecular genetics and paved the way for techniques that would allow scientists to manipulate and study genes in unprecedented detail. The central dogma in place, the field of molecular genetics blossomed. Scientists developed new tools to analyze DNA, RNA, and proteins, leading to discoveries about gene regulation, mutation, and expression. Techniques like DNA sequencing and recombinant DNA technology emerged, allowing researchers to read and manipulate genetic material. These advances laid the groundwork for the birth of genomics as a distinct scientific discipline. In the 1970s, scientists developed the first methods for sequencing DNA. Frederick Sanders' chain termination method and the Maxim Gilbert technique made it possible to determine the order of nucleotides in DNA. These pioneering methods were labor-intensive but represented a monumental step forward. For the first time, researchers could read the genetic code directly, opening new possibilities for understanding genes and genome. Development of recombinant DNA technology in the 1970s allowed scientists to cut and paste DNA from different sources. This breakthrough enabled the creation of genetically modified organisms and the production of important medicines like insulin. Recombinant DNA technology also made it possible to clone genes and study their functions in detail, marking the beginning of genetic engineering and biotechnology. 1983, Kary Mullis invented the polymerase chain reaction, or PCR. This technique allows scientists to make millions of copies of a specific DNA segment quickly and accurately. PCR revolutionized molecular biology by making it easier to study genes, diagnose diseases, and conduct forensic analyses. Its impact on genomics has been profound, enabling large-scale genetic studies and the rapid detection of genetic variation. In the late 1980s, the term genomics was coined to describe the comprehensive study of entire genomes. Unlike genetics, which often focuses on individual genes, genomics looks at all the genetic material in an organism. This shift in perspective allowed scientists to explore the complexity of life on a much larger scale, leading to new insights into evolution, development, and disease. 1990, the Human Genome Project was launched as an international effort to map and sequence the entire human genome. This ambitious project brought together scientists from around the world, aiming to identify all the genes in human DNA and understand their functions. The Human Genome Project marked a new era in biology, promising to transform medicine, agriculture, and our understanding of human evolution. Human Genome Project used advanced sequencing technologies to read the 3 billion base pairs of human DNA. The process took over a decade and required massive computational power. By 2003, the project was completed, providing the first complete map of the human genome. This achievement was a milestone in science, offering a blueprint for understanding health, disease, and human diversity. Completion of the Human Genome Project transformed medicine by enabling personalized approaches to diagnosis and treatment. Doctors can now use genetic information to predict disease risk, tailor therapies, and develop new drugs. Genomics has also improved our understanding of rare diseases, cancer, and inherited conditions, leading to more effective and targeted healthcare solutions. Genomics has also revolutionized agriculture. By analyzing the genomes of crops and livestock, scientists can breed plants and animals with desirable traits, such as disease resistance or higher yields. Genomic technologies have made it possible to develop genetically modified organisms that are more nutritious and sustainable, helping to address global food security challenges. Study of genomes has provided new insights into evolution and the relationship between species. By comparing the genomes of different organisms, scientists can trace evolutionary lineages, identify shared genes, and uncover the genetic basis of adaptation. Genomics has deepened our understanding of how life on Earth has evolved and diversified over millions of years. Development of next-generation sequencing (NGS) technologies in the 2000s made it possible to sequence DNA faster, cheaper, and more accurately than ever before. NGS has democratized genomics, allowing researchers and clinicians to analyze entire genomes in days instead of years. This technological leap has accelerated discoveries in medicine, biology, and beyond. With the advent of affordable sequencing, personal genomics has become a reality. Companies now offer direct-to-consumer genetic testing, allowing individuals to learn about their ancestry, health risks, and traits. Personal genomics empowers people to make informed decisions about their health and lifestyle, but it also raises important questions about privacy and ethics. Epigenomics is the study of chemical modifications to DNA and its associated proteins that affect gene expression without changing the DNA sequence. These modifications can be influenced by environment, lifestyle, and disease. Epigenomics has revealed new layers of complexity in gene regulation and has important implications for understanding development, aging, and cancer. Etigenomics involves the study of genetic material recovered directly from environmental samples, such as soil, water, or the human gut. This approach allows scientists to explore the diversity of microbial communities and their roles in ecosystems and human health. Metagenomics has led to the discovery of countless new species and has expanded our understanding of the microbiome. Functional genomics seeks to understand how genes and their products interact to produce the traits and behaviors of organisms. By using high-throughput techniques, scientists can study gene expression, protein interactions, and metabolic pathways on a genome-wide scale. This field bridges the gap between genetic information and biological function, offering insights into health, disease, and evolution. Comparative genomics involves comparing the genomes of different species to identify similarities and differences. This approach helps scientists understand evolutionary relationships, gene function, and the genetic basis of traits. Comparative genomics has revealed the deep connections between all forms of life and has provided clues to the origins of complex traits and diseases. Genomics has transformed cancer research by enabling the identification of genetic mutations that drive tumor growth. By sequencing the genomes of cancer cells, scientists can develop targeted therapies that attack specific mutations. This personalized approach has improved outcomes for many patients and is paving the way for new treatments and early detection methods. Pharmacogenomics studies how genetic differences affect individual responses to drugs. By understanding these variations, doctors can prescribe medications that are more effective and have fewer side effects. Pharmacogenomics is a key component of personalized medicine, ensuring that treatments are tailored to each person's unique genetic makeup. As genomics advances, it raises important ethical, legal, and social questions. Issues such as genetic privacy, discrimination, and informed consent must be carefully considered. Policymakers, scientists, and the public are working together to ensure that the benefits of genomics are realized while protecting individual rights and promoting responsible use of genetic information. The explosion of genomic data has created new challenges and opportunities in data science. Bioinformatics combines biology, computer science, and mathematics to analyze and interpret vast amounts of genetic information. Advances in artificial intelligence and machine learning are helping scientists uncover patterns and make predictions that were previously impossible. Discovery of CRISPR-Cas9 technology has revolutionized genome editing. This tool allows scientists to make precise changes to DNA, opening up possibilities for treating genetic diseases, improving crops, and studying gene function. CRISPR has made genome editing faster, cheaper, and more accessible, but it also raises important ethical questions about the limits of genetic modification. Single-cell genomics allows scientists to study the genetic material of individual cells. This approach has revealed the incredible diversity of cell types and states within tissues and organisms. Single-cell techniques are helping researchers understand development, disease progression, and the complexity of biological systems at an unprecedented level of detail. Population genomics examines the genetic variation within and between populations. By studying large groups of individuals, scientists can identify genetic factors that contribute to health, disease, and adaptation. Population genomics is essential for understanding human diversity and for developing public health strategies tailored to different communities. Genomics has played a crucial role in understanding and combating infectious diseases. By sequencing the genomes of viruses and bacteria, scientists can track outbreaks, identify mutations, and develop vaccines. Genomic surveillance has been especially important during pandemics, enabling rapid responses and informed public health decisions. Many rare diseases are caused by genetic mutations. Genomic sequencing has made it possible to diagnose these conditions more accurately and quickly. By identifying the genetic basis of rare diseases, researchers can develop targeted treatments and provide hope to patients and families who previously had few answers. Pharmacogenomics is increasingly being used in clinical practice. Doctors can now use genetic tests to determine which medications and dosages are best for individual patients. This approach reduces the risk of adverse drug reactions and improves treatment outcomes, making healthcare more precise and effective. Genomic analysis has opened new doors for exploring human ancestry and migration. By comparing genetic markers across populations, scientists can trace the movements of ancient peoples and uncover the roots of modern populations. Ancestry testing has become popular, allowing individuals to connect with their heritage and learn about their family history. Conservation genomics uses genetic information to protect endangered species and preserve biodiversity. By analyzing the genomes of threatened animals and plants, scientists can identify genetic diversity, track populations, and design effective conservation strategies. Genomics is helping to safeguard the future of our planet's ecosystems. Synthetic genomics involves designing and constructing new genetic sequences from scratch. Scientists can create artificial genes, chromosomes, or even entire genomes, opening up possibilities for new medicines, biofuels, and materials. This emerging field blurs the line between biology and engineering, offering exciting opportunities and challenges. Human microbiome, the collection of microbes living in and on our bodies, has become a major focus of genomics research. By sequencing microbial genomes, scientists are uncovering the roles these organisms play in health, digestion, immunity, and disease. Understanding the microbiome is leading to new approaches in medicine and nutrition. Genomics has transformed forensic science by enabling the identification of individuals from tiny DNA samples. DNA profiling is now a standard tool in criminal investigations, disaster victim identification, and paternity testing. The accuracy and reliability of genomic techniques have made them invaluable in the pursuit of justice. Researchers are using genomics to unravel the mysteries of aging. By studying the genetic and epigenetic changes that occur over time, scientists hope to understand why we age and how to promote healthy longevity. Genomics is shedding light on age-related diseases and potential interventions to extend healthy lifespan. Genomics is helping to uncover the genetic factors that contribute to mental health conditions such as depression, schizophrenia, and autism. By identifying risk genes and understanding their effects, researchers are developing new approaches to diagnosis, treatment, and prevention. Genomics is bringing hope to millions affected by mental health challenges. Nutrigenomics studies how our genes interact with the foods we eat. By understanding these interactions, scientists can develop personalized nutrition plans that optimize health and prevent disease. Genomics is revealing why people respond differently to diets and how nutrition can be tailored to individual genetic profiles. Environmental genomics examines how genes interact with environmental factors such as pollution, toxins, and climate change. By studying these interactions, scientists can assess risks, develop interventions, and protect public health. Genomics is providing new tools for understanding and addressing environmental challenges. Artificial intelligence is playing an increasingly important role in genomics. Machine learning algorithms can analyze massive genomic datasets, identify patterns, and make predictions about gene function and disease risk. The synergy between AI and genomics is accelerating discoveries and transforming the way we approach biological research. Future of genomics is full of promise. Advances in technology, data analysis, and our understanding of biology are opening new frontiers in medicine, agriculture, and environmental science. As costs continue to fall and knowledge expands, genomics will become an even more integral part of our lives, shaping the way we understand and improve the world around us. Despite its many successes, genomics faces significant challenges. Issues such as data privacy, ethical dilemmas, and unequal access to technology must be addressed. Continued collaboration between scientists, policymakers, and the public will be essential to ensure that the benefits of genomics are shared by all and that potential risks are managed responsibly. As genomics becomes more important in society, education is key. Schools and universities are incorporating genomics into their curricula, preparing the next generation of scientists, healthcare professionals, and informed citizens. Public outreach and education initiatives are helping people understand the opportunities and challenges of this rapidly evolving field. History of genomics is a story of discovery, innovation, and transformation. From the earliest theories of heredity to the sequencing of entire genomes, genomics has reshaped our understanding of life. As we look to the future, the potential of genomics to improve health, solve global challenges, and deepen our knowledge of the natural world is greater than ever. The genomic revolution is just beginning, and its impact will be felt for generations to come.